right, good morning, everyone. I'm Jason Moore. I'm chair of the Department of Computational Biomedicine at Cedars-Sinai Medical Center, which is about a 15-minute drive east of here. I also have an adjunct appointment in the Department of Biostatistics here at UCLA. I hope you all are having a great week. It looks like a wonderful program. So I'm a computational geneticist and want to tell you a little bit about a new method we've developed for automated quantitative trait locus analysis. So let's start with genetic architecture. As we think about the genetics of a complex trait like coronary artery disease that I have shown here, we ask questions about how many genes are involved, what are the variation in those genes and or around those genes, and what are the effects of those variations on the phenotype that we're interested in. And the point of this slide is to say that a disease like coronary artery disease is very complex. It's probably hundreds of genes with many thousands of genetic variants that code for proteins that are involved in a variety of different biochemical systems like lipid metabolism, carbohydrate metabolism. These systems interact with each other, and all this biology happens in the context of our environmental exposure, and that's ultimately what determines our trajectory toward a clinical endpoint such as stenosis of your coronary artery. I'm going to talk today about quantitative traits. This figure is looking at the distribution of LDL cholesterol among low risk, high risk, and moderate risk patients. This is about 11,000 subjects from a Swiss study. These subjects are all on statins, cholesterol lowering medication, but the point of this slide is to look at the variability of LDL cholesterol. And we can ask the question of what contributes to that variability. Obviously, diet's going to play a big role, smoking and other environmental exposures, how much you exercise, the medications you're taking. These individuals are on statins, so they are taking a medication that's impacting their cholesterol levels, their weight, their height, etc. Stress, of course, plays a role, and genes play a role as well. And so the goal of quantitative genetics is to understand how much of the variability in cholesterol levels among individuals is due to genetic variation. And so to do that, we employ what's called quantitative trait locus analysis. And I'm going to talk specifically about data coming from a study of rats. And so I have here on the left of this slide a study design for mice. That's commonly used. This is a study design that took 8 different mice with different genetic backgrounds and crossed them in different ways to create a resource of variability among mice that could be used to carry out quantitative trait locus analysis. So you have a quantitative trait of interest, you measure genetic variation across the mouse genome, and then you look at each SNP, each single nucleotide polymorphism, and its relationship with mean levels of the trait that you're interested in. So that's shown here in these upper right panels. So here is a SNP with 3 genotypes, and we see a mean change in the quantitative trait depending on genotype. And then the one on the right, there's no difference. So we call this SNP2, we call a QTL, a quantitative trait locus, and SNP1 is not a QTL. And we do this across the human genome and correct for the number of multiple tests that we do. That's shown in the red line here. And then the ones above the line are the ones that are considered statistically significant and considered QTL. So this is the standard way of doing this, and the analytic approach that's used is a parametric linear regression, regressing the genotype values on the quantitative trait. And then just as a matter of background, a more focused type of quantitative trait locus analysis is expression quantitative trait locus analysis, where you're actually treating gene expression levels as the quantitative trait of interest and looking across the genome to find QTLs associated with expression levels. So same concept, just a different measured quantitative trait. Now, as I said, parametric linear regression is the workhorse of quantitative trait locus analysis. This is what everybody does, and there are a lot of good reasons to use this analytic approach. The mathematical theory's been around for a long time and is very well understood. Linear regression is computationally efficient, right? We can carry out a genome-wide analysis with 1 million SNPs in no time. You could do that on your laptop computer. There's a deep literature base because this is what everybody does. There's thousands of papers on QTL analysis with linear regression for you to study and draw on. And the results are easy to interpret, especially because we're often doing a univariate analysis of one SNP and one quantitative trait, and looking at the additive relationship among the different genotypes on that trait. Now, there are some disadvantages, and let me say that the goal of regression is to do inference, right? We're controlling the data in a certain way. We're using this mathematical theory to test hypotheses, and because everything's controlled, we can make an inference about what we believe is real, and there are a lot of advantages to that. Now, the disadvantage is that there's a lot of assumptions that go into linear regression, and if some of those assumptions are not true, then the results might not be valid or might be skewed in some way. Parametric linear regression is powered to detect the individual univariate effects of a SNP, but not so much more complex, say, interactions between multiple SNPs or SNPs and environmental factors. And also, when you do this on a genome-wide scale, you're paying to do inference, to care about false positives, we correct for multiple testing, and that can reduce our power. Some valid SNPs will be missed just because we're applying a very stringent significance threshold because we've done so many tests. And as I've said, this ignores the complexity of the problem that I showed in my first slide. Many genes, many environmental factors working together in a complex way to determine the variability in the quantitative trait that you're interested in. Now, I would never tell anybody not to do linear regression. I think this is a very appropriate thing to do with quantitative trait data. But what I want to spend the rest of the time talking about is how you approach this problem from a machine learning point of view. And I would say that both approaches complement each other. Yes, you want to do statistical inference using all of the power of parametric statistical approaches, and that's a good thing to do, and you get a lot of benefit from that. The reason we do machine learning is to find the things that linear regression misses. And so I would argue that the two approaches are complementary, but looking at the data in a very different way. And again, regression's doing inference, and machine learning, what we're doing is prediction. It's a very, very different approach. Now, machine learning has advantages. It's going to embrace the complexity. We can look at multiple SNPs simultaneously. We could throw in environmental factors, other things that you think are important. And it's going to be able to model the interactions, the complexity, the heterogeneity of those genetic effects. There are fewer assumptions. And as I said, we're really focused on prediction, which is a very different problem than statistical inference. Now, of course, there are disadvantages to machine learning. We worry about overfitting the data, learning the noise in the data. It can be hard to implement, and that's going to be really the focus of my presentation today, is how can we make machine learning easy so that absolutely everybody can do it regardless of your computational background. It can be computationally expensive, requiring high-performance computing. Machine learning models are harder to interpret and perhaps less actionable if your ultimate goal is to biologically validate the SNPs that you're finding. So just a quick review of what machine learning is for those of you that are not familiar with machine learning. Here on this slide, I love this slide. You can find this online. I have the web address here. I did not make this, but it's readily available on the web. Shown on the left here are 3 different data sets, each with an X and a Y variable. So you could think of that as, say, body mass index and cholesterol, and the red dots are people with, say, cardiovascular disease, and the blue dots are healthy controls. And across each column are different commonly used machine learning models. And what you can see is what machine learning is trying to do is find a mathematical function that discriminates the red and the blue dots the best. And you can see that each of these machine learning models looks at the data in a different way. And the numbers represent the performance, like the predictive accuracy. The higher the number, the better. And you can see there's quite a bit of heterogeneity among the different methods and even across the different data sets. On some data sets, one method might be the best. On the next data set, it might be the worst. And so the point why I'm showing this slide is that this is one of the big challenges of machine learning, is that before you start your data analysis, you don't know what the right method is to choose. There's quite a bit of variability, and it's very much data set dependent. So my goal, and the goal of my research program over the last almost 10 years, has been how can we make machine learning easy and accessible to avoid some of these complexities? And I think this is one of the most intimidating things about machine learning is there are just so many decisions that need to be made. There are so many different algorithms that could be employed, and how you choose those and how you piece them together to do the most powerful analysis you can do is quite a challenge. So my goal is to make this easy and put this sophisticated technology in the hands of absolutely everybody. And we're going to do this specifically for this particular type of genetic analysis today. So the way we're going to do this is through automation. We're going to let the computer do all the hard work for you and make all the decisions for you and learn what the right way to do the machine learning is so that you don't have to do it. Okay, so as I've just said, there are a lot of decisions to be made. Let me review a couple of them here, just and specifically in the context of QTL analysis. So the first thing is, what data do you use? And you could have different data sets. Maybe you've processed them differently. Maybe you take all your SNPs from your genome-wide association study and parse them into pathways. And so what data do you use? What data cleaning methods or pre-processing methods do you use? Then you have to pick a machine learning algorithm. And as I said, there are dozens of different machine learning algorithms, each with strengths and weaknesses. You might want to do what's called feature engineering, where we combine features in a way that might make statistical or biological sense, or might make it easier for the machine learning algorithm to find a pattern. I've listed just some simple ones here, like you could add SNPs together or multiply them together or use a relational operator like less than or greater than. And that produces new features, new constructs that we can then use for machine learning. You might want to do feature selection. That was mentioned in the previous presentation. And in genetic studies, we often have 1 million or more genetic features, genetic variables to work with, and that can be very computationally intensive for machine learning. So you might want to select a subset, and there are lots of different ways to do that. You can do that based on allele frequency, select those SNPs that are more common. Based on gene annotation, maybe you just want to focus on the regulatory regions of genes, the enhancer and promoter of a gene. That's a very reasonable thing to do. Maybe focus on certain biochemical pathways. If you're studying heart disease, you might want to focus on cholesterol metabolism or blood pressure regulation and biological function. So there are a lot of decisions that could go into selecting subsets of features to use. And then the data doesn't always come to us coded in the right way. Now, in a standard QTL analysis, the additive encoding, where you code the genotype 0, 1, and 2 based on allele, the number of alleles of a particular type of allele is what's typically done. But there are other types of genetic effects, dominant and recessive effects, for example, and that requires a different type of genotype encoding. And that's not typically done when you do linear regression, because you pay a multiple testing penalty for every variation on the analysis that you have to consider. But in machine learning, this could play a very important role, because if you code the data in a way that makes it easier to detect the signal, that's work the machine learning algorithm doesn't have to do. But again, this requires decisions. So there are a lot of decisions here. Some are machine learning decisions, some are genetic analysis decisions, and my goal is to automate all of this so the computer figures out how to make all these decisions. So here's conceptually what we're doing, and our method is called automated quantitative trait locus analysis. So here we have the computer represented by this robot arm here. So we don't actually use a robot arm, but conceptually you have the computer picking data sets, machine learning algorithms, feature engineering algorithms, feature selection algorithms, feature encoding algorithms, and piecing these together in a pipeline to do an analysis. And it considers lots of different combinations of pipelines, types of pipelines. It executes them on your server. We get a model back, and then the computer evaluates those models. And then the key here is it makes variation in the pipelines. It generates new pipelines based on the performance of the pipelines it's evaluated, and then iterates this cycle until it gets a high-performing pipeline that you can then take and do your analysis with. So this is conceptually how AutoQTL works. Let me just step through the basic components. The first thing we need is a machine learning code base. We use the scikit-learn library for this, which, if you're not familiar, is one of the most commonly used open source Python-based libraries. It does pretty much everything in machine learning that you want it to do, except maybe deep learning. So we use this as our code base. When we need a machine learning algorithm, it pulls from this library and executes it. Second thing we need is a way to represent machine learning pipelines in the computer so they can be easily manipulated and scored. And we use a type of data structure in computer science that's called an expression tree. So here at the root of this tree, we have a machine learning method that's doing regression, something like, it could be linear regression, it could be a regression tree or a regression neural network. And then that machine learning method has a set of parameters that are tuned here by the method. In this particular pipeline, the next operator here, it's doing a feature engineering step. It's multiplying the values of this SNP x1 times the processed values of x2 and x3 here. So we're doing some feature engineering. The next level down in the pipeline, it's doing feature selection. So it's selecting based on gene annotation here among the set of SNPs that are fed into it. And then at the lowest level, this is the beginning of the pipeline here, SNP3 is receiving a dominant encoding. And then between x2 and dominant x3, they're being selected based on gene annotation multiplied times x1. That gets fed into the neural network, which then is predicting the values of the quantitative trait that you're interested in. So what the algorithm does is it's generating hundreds or thousands of pipelines like this. They can be any shape, they can be any size, they can be any combination of operators. We do something called genetic programming, which is really ideally suited to this expression tree representation, which is a pretty simple idea. We randomly initialize hundreds or thousands of these pipelines. We evaluate them. We select the best ones based on some criteria, and I'll show you what we do in a minute. Then we do this crossover mutation operator, which is basically just swapping different parts of the expression trees to generate new expression trees. And then you iterate through this variability evaluation selection algorithm until you reach some stopping criteria. Now the key here is how we select and evaluate these pipelines. And one of the things we can worry about a lot in machine learning is overfitting, learning the noise in the data such that the model won't generalize to new data. And so we do something called, we do cross-validation, which is dividing the data into training and testing pieces, but we also do something called Pareto Optimization, which I think plays, for this particular application, plays a very important role. And this is a type of what's called multi-objective optimization. So the machine learning is being, the pipelines are being evaluated based on multiple criteria. And here we're measuring the variation explained by a particular machine learning model by R squared. And we have training and testing data. So we split the data into two different pieces. And so on the x-axis here, we're evaluating the predictive ability of the model. How good of a job is it doing at predicting values in data it hasn't seen yet? And then on the y-axis, we're looking at a function of the difference between the training and the testing R squared. And that's important because that's a measure of overfitting. So if you're overfitting on the training data and have a high R squared, and you're learning the noise in the data, it's not going to generalize to the testing data that you've held out, and that R squared should be much lower. And so that training testing difference is a good measure of how much your algorithm is overfitting. And so in Pareto optimization, we're balancing those two things. Yes, we want a model that predicts well on the x-axis, but we also want a model that's not overfitting on the y-axis. And so here, what we do is we generate hundreds or thousands of pipelines. We put them in this two-dimensional space, and we identify what are called the Pareto optimal models here on the blue line, this outer edge. These are the models that best balance these two factors. So here, up in the upper left, we have a pipeline that is not overfitting very much. So that's good, but its predictive ability is not very good. So that's perhaps not a very interesting model. Here's one that predicts really well, but seems to be overfitting quite a bit. And so we typically look at this bend here for pipelines that balance these two objectives, and that's where we look for our best models. So this is a new method. We've just developed it in the last year. We have a paper that just came out in Biodata Mining, which is an open access journal. This is work done by Freda, my postdoc, and Ghosh A, my data scientist. So to evaluate this method, we applied it to a rat GWAS data for body mass index. And this is work, our collaborator Abraham Palmer is one of the senior authors on the study. He's down at UC San Diego. So this is over 3,000 outbred rats, and they had a number of different body weight type measurements and did a genome-wide association study using QTL analysis as I've described. This is a Manhattan plot looking at the statistically significant hits they got across these different traits. And we asked them for what they thought the best hits that they got, and they sent us the SNP IDs for 18 SNPs from this study. And those are the 18 that we put into AutoQTL to see what it should find. Now let me say that when you do a GWAS, univariate linear regression is designed really to find those SNPs that have independent effects regardless of genetic background. So that's really what you're finding. So when we run AutoQTL on these 18 SNPs detected using linear regression, we would hope that AutoQTL would find linear regression as the best method, and that's what the pipelines should represent. Here is the Pareto optimal models that we found here. And again, we kind of look at this bend here, this middle part as a balance between the difference and the testing R-squareds. And these are the Shapley values. Think of these as for each of the 18 GWAS hit SNPs, this is the relative importance of each SNP to the predictive ability of the pipeline. And I want to call your attention to this red asterisk here. This model was selected automatically by AutoQTL, and it's an example where if you take all 18 SNPs and put them into linear regression, that's what you get. Note that it's very high on the testing prediction, which is good, but it seems to be overfitting because it has a very low difference score. So that suggests that these 18 SNPs are, for predictive purposes, we're not talking about inference here, but for predictive purposes might be overfitting the data and learning noise and not generalizing. Yeah?
Sorry, I missed your definition of the difference score.
So if you calculate the R-squared on the training data that the model is trained on, and then you calculate the R-squared with the model fixed on the testing data, the holdout data, what's that difference? So it's a function of the difference. It's actually 1 over the absolute difference, and we take a root square of that to adjust the scale. But basically, the higher the value on that y-axis, the less difference there is between the training and testing score, and the less overfitting that's happening. The bigger the difference between the training and testing, the more the model is learning the training data, not generalizing to the test data.
So this is under the assumption that these 19 SNPs are the causal SNPs, but how does this account for any of these structure?
So they did some validation in their paper, so some of these were validated, but yeah, these 18 are based on inference, right? Genome-wide correction for multiple testing, statistical inference, and they may not all be valid functional SNPs or tagging valid functional SNPs nearby. And our results suggest that might be true, that it really, and I'll show you on the next slide, it really seems to be a subset of these SNPs that is providing the predictive ability of the model and not all of them. When you put all of them in a regression model, they seem to overfit when you do prediction. Yeah.
Just a question for clarification. If your collaborators gave you these 18 SNPs, were they from the entire discovery sample?
Yeah, from the whole, the entire GWAS, yep.
So how can you then do cross-validation because it's not independent anymore?
Yeah, that's a good question. I think the difference here is that they were doing inference on those SNPs, and we're putting them into a machine learning model and doing prediction. But you're right, there might be a little bit of, but that would argue, your point would argue the other way, that if that was an issue, which I agree it could be, but if it was an issue, then it would create more false positives rather than fewer false positives. Right? Our machine learning model would be more likely to be finding false positives based on that. But it's a good point. Okay, so let me move on. So here are what the pipelines look like, and we've just represented them as a table here. So here's the first operator in the pipeline, second, third. And you can see for these two, the bolded ones are the two arrows here in the middle, the ones that I say balance the two criteria. Basically what AutoQTL is doing is it's building a pipeline where it's doing, the VT is a variance threshold, so it's doing a feature selection and it's doing a feature encoding using a recessive encoding, and then combining that with linear regression. And these are the two that balance the objectives the best. Linear regression, and it did select linear regression with all 18 SNPs here, down here, and these two models, these two down here at the bottom that have the high testing R-squared, but seem to be overfitting. So these two in the middle here are doing a selection, something like half the SNPs are selected, fed into linear regression, encoded with a recessive encoding, and that seems to balance the overfitting and the predictive accuracy. Now, the point I want to make is that we didn't tell AutoQTL to do linear regression. It had access to multiple machine learning methods. It had access to all the things in the library, and it automatically selected linear regression and automatically pieced that together with genotype encoding and feature selection strategies to improve the overfitting of the model. Now, the next question we had was, okay, well, that's great. That convinced us that it's doing what we think it's doing. But what happens if we feed it data where there are interactions? And we simulated a set of data where, with the same number of SNPs, 18 SNPs, where each pair of SNPs is involved in an epistatic interaction, a non-additive interaction, such that if you do univariate linear regression, you shouldn't get much predictive accuracy, and you need machine learning to be able to detect those patterns. So when we ran AutoQTL, it automatically discovered that machine learning algorithms perform the best. And this is just showing over hundreds of runs that it's picking random forest and decision trees preferentially over linear regression. And down here, it's picking different feature encoding, recessive dominant type encoding strategies over the additive encoding. So when you give it epistatic effects, it's doing its job. It's not picking linear regression. It's picking the machine learning methods automatically. And here are what the pipelines look like at the bend here of the Pareto front. And you can see it's doing a lot of coding here, a lot of dominant encoding, and then combining that with random forest or decision trees here. So these are the models here in the balance of the two training objectives. So, and again, it's doing this completely automatically without human input. So when it's presented with a data set with additive effects, it's picking linear regression. When it's presented with a data set with non-additive effects, it's picking machine learning methods, and it's doing that automatically. All right, so I'll finish up. And obviously, this is just the beginning. And as I said earlier, machine learning models make all of this difficult to interpret. But there are some things you can do. On the biology side, you could certainly look at gene co-expression of the things that it's finding in the model, gene set enrichment analysis, species comparison, et cetera. On the computational side, we can look at the feature importance scores. I showed Shapley values earlier, which are built into the method. And then you could build a prioritized list of SNPs that you might then experimentally validate using something like base editing, where you go in and change the genotypes in a way that allows you to test the non-additive interaction that the machine learning is finding. We have a review paper on automated machine learning approaches for genetics and genomics. This is the first such paper published. This was just published last year in Human Genetics. And let me thank Phil Freda and Autry Ghosh, who did all the work on AutoQTL. my email address, so if we can be helpful, feel free to reach out. AutoQTL is open source. It's all Python-based. It's on GitHub. You can download it and use it. And if you have any questions, let me know. And this is the Pacific Design Center in West Hollywood, where our offices are located. This is expansion space for Cedars-Sinai Medical Center, which is about a 10-minute walk away. So this is, we're located on the 5th floor of the green building here. All right. Hopefully there's time for a few questions.